So hi everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry, and in today's session we are going to discuss about replication of DNA. Okay, so what do you mean by DNA replication? What is the purpose of replication of DNA? So you create from a parent strand of DNA, you create two identical copies of daughter strands. That is the purpose of DNA replication. Why do you want to replicate your DNA? Why do you want to make two daughter copies of the same parent strand? Because once the DNA replicates, then only the entire cell can replicate, and cell replication is extremely important. Cells can multiply. Cells are not replicating, cells can multiply, and the cell multiplication is extremely important for our growth and all other physiological functions. So DNA replication gains a very important significance in this context, and this process is a semi-conservative method. What do you mean by semi-conservation? See, we have got a parent DNA here. This is a parent DNA. So a parent DNA has got two strands because it is a double-stranded DNA, right? So it has got two strands. From the parent DNA, you synthesize two daughter DNAs. So among these two daughter DNAs, one half of this daughter DNA will be a original parent DNA strand. Okay, this will be the newly synthesized strand of the daughter DNA. Okay, so similar here, this is the parent one, this is the new strand. Okay, so it is a semi-conservative method. It means what? You have preserved half of the parent DNA strand into the newly synthesized daughter strand. Okay, so hence this process is called as a semi-conservative method. Are you clear? So this is the main purpose of DNA replication, and DNA replication, the main property is its semi-conservativeness. Now we move on into the important part of replication, which is the direction of replication. So you all should be remembering, listen to me very carefully, the direction of replication in a eukaryotic DNA or a prokaryotic DNA or whatever it is bidirectional. It means what? It can replicate on both the sides. That is called as bidirectional. Okay, and how the synthesis, how the ends will be there? I mean, the synthesis of new strand will be always having a polarity from 5' end to 3' end. Okay, the newly formed daughter strand will always be synthesized in this direction from 5' polarity to 3' polarity, and it is not the other way around. Please remember, this is an extremely important point in order for you to understand the entire process of replication. Clear? So always the synthesis direction is from 5' end to 3' end, but this is bidirectional. So how can this be following the same path, but it could be bidirectional at the same time? Because both the strands are complementary and anti-parallel to each other. For example, the original parent strand, say for example, this is the original parent strand. This original parent strand, for example, if this is the polarity of the first strand, okay, and if this is the polarity of the first strand, it means the polarity of the second strand will be how? 3' to 5'. So this is the double strand. Okay, this is anti-parallel, and if you are synthesizing a daughter strand, okay, the daughter strand will be synthesized in this direction only. That is, in case of this first strand, it will be synthesized in this direction, because here it is the 5' end and this is the 3' end. Whereas in this case, the new strand will be synthesized in this direction, wherein this becomes the 5' end and this becomes the 3' end. So it is bidirectional, but it always follows this rule of polarity from 5' to 3' it synthesizes. Okay. So this is the important point you are supposed to remember in case of replication. In case of reading any kind of an RNA primer or DNA polymerases, they read in 3' to 5' direction, but they synthesize the original product from 5' to 3' direction. This is about reading of parent strand, and this is about synthesis of daughter strand. There is a specific reason for following this kind of a direction. I will explain in a short while now why this direction is being maintained in case of various kinds of DNA polymerases. Now, with this, we move on into the starting of replication, that is initiation of replication.